Good morning, good afternoon, everyone. This is Mahalea Marshall from the IQ Consortium Secretariat. I'd like to thank you for joining this morning's IQ webinar on the development of Ritonavir Pediatric Formulations, presented by the IQ Drug Product Pediatric Formulations Working Group. This webinar will be recorded and will be available via the IQ Consortium website, as well as via distribution by today's presenting working group. Before I begin and turn things over to our presenters, I would like to take care of just a few housekeeping items. First, this webinar and presentation and subsequent Q&A session will conclude no later than 12 p.m. Eastern Time. Second, on the panel on the right of your screen, you will see a green arrow shown here. This green box will allow you to collapse or expand the panel. Next, voice over internet protocol participants can avoid audio feedback by using a headset and keeping wireless devices away from their computers. For those calling in by telephone, please enter the audio PIN number here, which will allow us to mute your line if we do experience feedback from your line. Finally, the Q&A session will take place at the conclusion of the presentation. Questions may be entered into the box highlighted here and will be read by the moderator during the Q&A session. Finally, I'd like to remind everyone that this webinar is subject to antitrust guidelines adopted by the IQ Consortium Board of Directors. These guidelines require us to stay away from discussion on any sensitive topics such as pricing or market shares. I'd like to now turn things over to Dr. David Tan from the DPLG Pediatric Formulations Working Group, who will begin our webinar session. David?
Thank you, Mahalia. So today I have the privilege to introduce our distinguished speaker, Dr. John Morris. So Dr. John Morris is a CMC Product Development Director at AbbVie with responsibility for CMC development of drug products. John received his bachelor's in chemistry from Augustana College in 1988 and a PhD in analytical chemistry from Iowa State University in 1993. So John joined Abbott in 1993, and he has led the CMC team for development, regulatory submissions, and launch of Abbott's first amorphous formulation, Kaletra, and recently guided a project team to deliver new technologies to enable acceleration of AbbVie's portfolio of HCV compounds. John has a development, global regulatory filing, and agency inspection experience for approximately 15 commercial products, including multiple fixed-dose combinations marketed in more than 170 countries. He was also sponsored by Gates Foundation to represent the pharmaceutical industry to present in a CMC point to consider for fixed-dose combination for HIV therapy at the WHO meeting in Geneva, Switzerland, on fixed-dose combinations for HIV to participants of WHO, pharma industry, FDA, UNICEF, MSF, and many other key opinion leaders. In 2010, John was inducted into AbbVie's prestigious Provider Society and named Outstanding Researcher. He has continued to contribute vastly to the company and was handsomely rewarded with the Chairman's Award and AbbVie Innovation Award. John has strong interest in amorphous solid dispersion, and today he will be sharing with us the development of Ritonavir pediatric formulation. And please welcome John Morris.
Thank you, David, and thank you for the opportunity to speak to you today on the development of Ritonavir pediatric formulations. In considering development of a new, improved Ritonavir pediatric formulation, this effort certainly is not starting from scratch. In fact, we have over 20 years of marketed product experience providing Ritonavir to HIV patients in a variety of formulation types. We know today that given the evolution of treatment paradigms for HIV patients, Ritonavir is no longer prescribed as a monotherapy or sole protease inhibitor regimen for patients. It is continued to be used in co-administration manner as a pharmacokinetic PK enhancer at low doses, where it capitalizes on the action of ritonavir to inhibit metabolism of the other protease inhibitors. This really allows the HIV protease inhibitors to maintain efficacy with reduced pill burden, dosing frequency, and food restrictions. This has enabled ritonavir to be a mainstay in the management of both treatment-naive and treatment-experienced patients. Under the paradigm where ritonavir as a PK enhancer is most commonly used at doses of 100 to 200 milligrams once or twice daily, according to approved labels of various HIV protease inhibitors. Ritonavir itself has always been a challenge to formulate. The molecule has very low water solubility in the low microgram per milliliter range. Solubility can be enhanced under very strongly acidic conditions to the low milligram per milliliter range at pH 1, but with the potential to really compromise chemical stability in that scenario. We knew from early preclinical studies that ritonavir showed essentially no bioavailability when administered in animal PK studies as an unformulated crystalline solid in a capsule. Incorporation of surfactants or acids or wetting agents, other excipients, really failed to increase the bioavailability to anything greater than small single digits of bioavailability. Unfortunately, similar results were also observed with salts of ritonavir that were explored. The low pKa really limits salt development, and in most instances, the solubility of the salt was really not adequate to overcome the low bioavailability observed for the parent molecule. We know in developing any active pharmaceutical ingredient, there's a number of factors that need to be taken into consideration to really achieve acceptable in vivo performance while demonstrating acceptable chemical and physical stability over shelf life with a product that is routinely manufactured by a robust and efficient manufacturing process. And it's important in particular for chronic administration for a very critical drug that we maintain a very consistent quality to ensure safety and efficacy to this patient population. So in this triangle of bioavailability, physical and chemical stability, and manufacturability, we have found through the years that often to make gains in one particular area, that actually often is confounded with giving up in another area. So there are a number of trade-offs. And we have found examples where in certain direction to optimize bioavailability to work with low drug loading or incorporation of surfactants, that often can compromise perhaps the physical and chemical stability, or even in other areas to result in a large number of dosage forms or large volumes to be administered. We know ritonavir having very low water solubility is further confounded with poor permeability. So in the BCS classification system, this is a class 4 molecule, and ritonavir has multiple polymorphic forms, in this case with drastically different physical-chemical properties. This makes it very difficult to formulate to reliably demonstrate acceptable and consistent bioavailability, and our ability to predict in vivo performance is very limited by establishment of in vitro laboratory tests or models. And this is really due to the very complex performance of ritonavir in the body. So in the ritonavir history, I mentioned over 20 years, there's actually 5 marketed formulations. This originated in the first products to be provided to patients in the mid-90s there was simultaneous launch of a Ritonavir oral solution and a Ritonavir hard gelatin capsule. These first formulations and Ritonavir itself were really the first of 3 protease inhibitors that became available in the mid-90s and really changed the landscape of treatment for HIV patients. Through ongoing product enhancement efforts, there was development of a Ritonavir soft gelatin capsule that was available to patients in 1999, and this was really served as a replacement for the original hard gelatin capsule. Further efforts sustained over the next 10 years were aimed at further enhancements and providing additional benefits to patients and caregivers. This resulted in a Ritonavir tablet formulation around 2009 as a replacement for the Ritonavir soft gelatin capsule. Continued efforts based on that success have now resulted in development and approval and launch of a Ritonavir powder formulation that has replaced the Ritonavir liquid in the EU and has introduced this powder formulation more recently in the US. With the original Ritonavir liquid formulation, those development efforts again played on the fact that we knew strong acids would help enhance solubility, but in fact were shown to definitely result in higher level of chemical degradation, so compromised stability. The solubility for Ritonavir is known to be enhanced in various organic solvents or in particular co-solvent mixtures, and this was utilized to maximize the drug loading and bioavailability. Ritonavir itself has a number of polar hydrophilic functional groups, but these are interspersed among nonpolar hydrophobic areas on the molecule and ultimately lead to the low water solubility. However, those features do provide opportunity with polar organic solvents or alcohols to have pharmaceutically acceptable organic solvents to lead to a liquid formulation. Despite having the drug completely dissolved in solution, it was observed that surfactants further enhanced the bioavailability and so were also incorporated into the formulation. This liquid formulation, based on finding the right balance of bioavailability, had to be refrigerated to minimize the degradation of Ritonavir over the entire shelf life of the compound or of the product. The hard gelatin capsule is a version that was a semi-solid formulation that contained a solidifying agent to minimize loss of alcohol and prevent leakage from the hard gelatin capsule. It was important to dissolve the Ritonavir in the alcohol co-solvent system. A pH modifier was included, and this was in an effort to achieve an acceptable drug loading to result in the 100 milligram strength capsule. Again, surfactants were incorporated to enhance bioavailability, and that was based on early preclinical studies. And the interesting feature for the manufacturer of this particular dosage form included a pre-dissolution step of Ritonavir in a hydroalcoholic solution, and this was mixed with a molten stream of the solidifying agent, maintained at an elevated temperature to allow mixing with the stream of solution containing the drug just prior to filling into the capsules. Based on the therapeutic dose of Ritonavir being 1,200 milligrams per day, this would require administration of a total of 12 100 milligram strength capsules. And again, this formulation with Ritonavir in solution was prone to chemical degradation and required refrigerated storage throughout the supply chain and throughout its shelf life. In 1998, there was a very unexpected event with the emergence of a new polymorphic form known as Form 2 that appeared throughout our drug substance and drug product manufacturing areas. Initially, during the early 90s through the mid-90s, all of the initial development work, there was only one known crystal form of Ritonavir, and polymorphism in general was not a major concern, since in all cases the drug substance is dissolved in the solution in the final product. With the emergence of Form 2, this crystal form exhibited quite different physical-chemical properties, and in particular, in the terms of solubility, exhibited about half the solubility compared to Form 1. This had a significant impact on the formulations, and the ethanol water solution utilized in the manufacture of Ritonavir capsules could not support the reduced solubility of Form 2, and it made this semi-solid formulation no longer viable, and that product had to be withdrawn from the market. In addition, there was also an impact on the Ritonavir liquid formulation. Similarly, a significant reduction of the Form 2 solubility in that solvent system. We had to also make modifications here, not to the formulation itself, but to the storage conditions. In this case, we know there's also a significant impact of temperature on the solubility of Ritonavir in these co-solvent systems. And to maintain adequate solubility in that oral solution product, we had to move the storage from refrigerated storage to room temperature storage in order to ensure that the Ritonavir remained dissolved in the formulation. This came at the expense of a significant reduction in the shelf life under those new storage conditions. So from a 2-year product at refrigerated storage, it now became a 6-month expiry under room temperature storage. So again, created a significant supply chain challenge to maintain that product and make it available to patients. In parallel to the unexpected work related to the Ritonavir new polymorph, there was really quite significant effort already underway, again, in entertaining additional product enhancement opportunities, in particular to improve patient compliance and offer a room temperature storage product for patient use. AbbVie had worked to develop a Ritonavir soft gelatin capsule formulation where now Ritonavir is dissolved in a co-solvent of a lipid, alcohol, and surfactant. This formulation was originally designed to maintain Ritonavir in solution at a higher concentration, a 200 milligram per gram concentration, and this was really based on the original crystal form, Form 1, and its solubility properties. Similar to the other marketed products, this product in development also was significantly impacted with the emergence and dominance of the now thermodynamically stable polymorph Form 2. And this required the soft gelatin capsule to be further modified to accommodate this reduced solubility for the new crystal form. Within a year of the emergence of Form 2, we were able to modify the soft gelatin capsule formulation and reduce the concentration to 100 milligram and launch this product in 1999 to provide, again, a solid oral dosage form to replace the original HTC formulation. So considering further product enhancement opportunities at this time, we recognize that there are multiple approaches that are applied to formulating drugs with low aqueous solubility. This includes particle size reduction approaches, things that are nanoparticles. It includes dissolving the drug in a lipid or a solvent with a surfactant, and this has been employed in our soft gelatin capsule formulation. And a third approach for drug delivery technology is converting the drug to a non-crystalline state or an amorphous solid dispersion. It was in this new area that we really had interest to continue to explore opportunities for product improvements that would provide additional benefits to patients. With any of these enabling formulation options, the objective is really to increase the dissolution rate and apparent solubility of the drug and ultimately result in improved enhanced bioavailability. However, each technology has opportunities and limitations, and often regarding drug loading, stability, and convenience factors. So our interest was really in further exploring the concept of amorphous solid dispersions to see if this would be appropriate or applicable for ritonavir. And there is evidence that in some cases for poorly soluble compounds, amorphous solid dispersions can achieve significantly higher plasma concentrations than with traditional formulations of crystalline solid forms. So in this case, this was enough interest for us to further pursue and really evaluate amorphous solid dispersions for ritonavir. And we knew, going back to our preclinical work, administration of ritonavir in a crystalline form really results in very low bioavailability. However, some proof of concept work with converting ritonavir to an amorphous non-crystalline form showed that significant benefits could be gained with this molecule. So in figure 2, there's a comparison of intrinsic dissolution rates of ritonavir comparing amorphous and crystalline forms. And you can see there's approximately a 10-fold advantage in intrinsic dissolution rate with the amorphous ritonavir. On the right-hand panel, there's an exploration of amorphous solid dispersion prototypes. In this case, these are PEG solid dispersions, comparing crystalline ritonavir to various drug-loading amorphous solid dispersions of ritonavir. And you can see there's a significant enhancement in this dog PK model with the amorphous solid dispersions. And there's even further evidence of an effect of the drug loading of the formulation, where the highest bioavailability comes with the lowest drug loading in that formulation. This early concept work really showed the potential, and we further identified hot melt extrusion as an appropriate manufacturing process to pursue further development and ultimately commercialization of a product. So what is an amorphous solid dispersion? We know with amorphous drug, this is a non-crystalline solid state composed of disordered arrangement of molecules, but that's not enough on its own. The amorphous solid dispersion is where the solute molecules are dispersed molecularly and within the amorphous solvent or carrier. On the right-hand side, the upper figure shows a elemental ritonavir sulfur map. So this is really selectively mapping sulfur, which is unique to the ritonavir molecule and is not present in any of the other formulation matrix excipients. And it really shows how uniformly the ritonavir is distributed throughout the entire matrix. Why is this important? It's really, again, to increase the solubility, but beyond the amorphous ritonavir benefits, it's really critical that the amorphous ritonavir is further stabilized within the amorphous carrier, the hydrophilic polymers, to maximize the surface area, avoid agglomeration, and improve the dissolution rate. This work ultimately led to development of a ritonavir 100 milligram tablet. So the experience gained with solid dispersion technology really showed that an ASD of ritonavir could achieve the desired bioavailability and show acceptable chemical and physical stability without refrigerated storage. The solid dispersion technology and the melt extrusion manufacturing process have been successfully employed to maintain the bioavailability observed with the liquid-filled soft gelatin capsule. And these benefits together for ritonavir tablets really translate through to benefits to the patient, caregivers, and it's primarily through the non-refrigerated storage that had been in existence for all of the prior ritonavir marketed product formulations. So why develop a new ritonavir pediatric formulation? So primarily this is really for considerations now for pediatric patients. All of the prior generations of product enhancements had provided significant benefits for adult patients, but throughout that time, the oral solution really remained for pediatric patients. We had a desire to develop a new formulation to replace this ritonavir liquid with a more suitable formulation for the pediatric population. In review here, some of the attributes of the current ritonavir liquid formulation, we know that this contains high level of alcohol solvents that can create a potential safety risk, especially in unfortunate situations with an overdose administration error. The required room temperature storage as a result of Form 2 for the liquid formulation significantly limited the shelf life, as mentioned, to 6 months under typical room storage conditions, room temperature storage conditions. And this really limited, ultimately, utility of this product in high temperature, high humidity climates. So looking at estimations provided by UNAIDS, and this is from 2015, HIV in the pediatric population continues to be a real global challenge. And you can see a distribution here. There still is estimated to be on the order of just under 2 million pediatric patients that would be classified under 15 years of age. There still is a significant unmet need. In comparing now to total numbers of people living with HIV, you can see that the population of pediatric patients under 2 million, but there are still significant numbers of deaths attributed to this. In 2015, still over 100,000 deaths. And alarmingly, there still are over 150,000 new infections that are occurring. When looked at this on a global basis, clearly there have been significant gains in providing HIV regimens and therapeutic options to patients on a global basis. But there still remains significant opportunity to be had. So in total, it's estimated that there are still approximately half of the existing pediatric patients that are still not finding access to treatment regimens for HIV. So in considering this, we developed some key design targets for development of the new ritonavir powder. The primary one was reduction or removal of the ethanol and propylene glycol solvents, again for safety considerations. Given that we are replacing an existing oral solution product, we needed to maintain flexibility to accommodate the broad range of doses that are applied to pediatric patients based on body weight or surface area. We want to ensure an acceptable dosage form for pediatric patients or even patients who may have difficulty swallowing the tablet. The storage stability we were really aimed at increasing our stability to achieve acceptable commercial shelf life under long-term storage conditions under global climatic conditions. It was critical that bioavailability needs to be comparable to what we had with the existing commercial oral solution. And of course, we wanted to explore opportunities to improve palatability for ritonavir. Our early formulation screening, one of the first concepts that we entertained was an enteric-coated powder. And this was primarily targeted at trying to have a formulation with increased taste masking to address the really inherent bitter taste of ritonavir that is well known from the Norvir oral solution, but still allowing the powder to dissolve in the intestine. The selection of the pH-sensitive polymeric coating was really to select one that is insoluble in acidic media, but soluble once it reaches the intestinal pH above 5.5. But it takes into account the various pH environments encountered throughout both the dose preparation as well as administration that could impact drug release and the associated taste attributes. Coated and uncoated powders were evaluated for ritonavir pharmacokinetics and palatability from aqueous suspensions. The result of those studies showed that comparable PK results could be obtained from coated and uncoated powder formulations. Bioavailability was in a range of 80 to 90% relative to the reference ritonavir liquid formulation. And palatability was compared across the coated and uncoated powders as well, and compared with that of the existing ritonavir liquid formulation. In this case, no taste masking benefit was really clearly observed from the coated powder particles. neither the coated or uncoated powders achieved improved palatability over the ritonavir liquid formulation. So in summary, the polymer coating did not impact the bioavailability, but also showed no apparent improvement in taste masking aspects. This was a little bit surprising to us, and we wanted to further investigate what could be the possible reasons for this. So upon closer examination, the powder that was used for this study had a rather broad particle size distribution and had a significant fraction of fine particles, less than 100 micron in size. In addition, the particle shape is not ideal for polymeric coating. We know for other conventional tablets, round pellets or mini tablets, this polymeric coating can be effective in achieving taste-neutral formulations. However, coating on these irregularly shaped particles did not result in a perfect barrier film coat. In fact, these particles are quite challenging to get a uniform coating, and upon very close examination, we found evidence of ritonavir drug particles that were still embedded in the outer polymeric coating, and being on the outer exposed surface of these coated particles would still lead to some of the off-putting taste aspects when administered. To further evaluate this approach, we produced a uniform 2 millimeter particle, and this size was actually selected based on reference to the FDA guidance for products labeled for sprinkle or small pediatric administration. There's guidance here that would target particle sizes up to 2.5 millimeter, and with a small allowance for sizes up to as large as 2.8. This is really gauged on providing a reasonable assurance that patients would be able to swallow the product with food without stimulating the urge to chew. So we produced compressed 2 millimeter particles with the same formulation and applied the same enteric polymeric coating to this. And in this case, unfortunately, now the coated particulates showed improved taste masking, but now at the expense of significantly reduced bioavailability relative to the ritonavir liquid formulation. It is believed that the delayed dissolution and drug release profile had a significant negative impact relative to a known very narrow absorption window for ritonavir in the upper intestine. The uncoated powder also provides a more homogeneous suspension than the coated powder, and this is also an important feature to ensure that accurate doses can be measured from an amorphous drug particle suspension for doses less than 100 milligram in pediatric patients. In addition to the bioavailability assessment and the enteric polymer coating assessment, it is important for us to really look critically at the chemical and physical stability for the amorphous solid dispersion concept. It's important that the ritonavir in the amorphous state is compatible with the formulation components and, importantly, maintains the desired solid form of the drug product over the entire shelf life and avoids any changes in the physical properties that could impact bioavailability. Stability was assessed in a commercial package over shelf life and under a wide variety of use conditions. This included long-term and accelerated stability in the commercial package configuration, bulk packaging, simulated in use if mixed with other food products for administration. And importantly, we have observed significant value in open dish studies that provided valuable information. The picture that's included here illustrates a microscopic observation when stressed under high relative humidity storage conditions where the formulation has really had significant uptake of water. This is a small fragment of a tablet of the ASD formulation, and what you can observe, the white spots that are distributed throughout the matrix are evidence of recrystallization of ritonavir, so conversion of amorphous ritonavir back to a crystalline form and we know what the impact could be to bioavailability under these conditions. A more systematic approach was applied to study the ritonavir powder in open dish conditions. And you can see under a variety of storage conditions with various relative humidities and various temperatures, these samples were observed over an extensive time period from, shown here, 6 weeks through 9 months. And at various test intervals, the samples were tested for water content. In some cases, TGs were determined, and we evaluated these applying near-infrared spectroscopy to look for evidence of crystalline ritonavir. You can see under very high-stress conditions, 75% RH and 40 degrees Celsius, these samples, the ritonavir powder has had significant water uptake, in some cases above 10% levels. You can see the effect on reducing the TG for this formulation. So now the TG at 27 degrees is lower than the storage condition. And these conditions together resulted in evidence of recrystallization occurring based on the NIR observations. You can see that initially these were crystalline ritonavir was detected at the 3-month test interval. Interestingly, both Form 1 and Form 2 crystalline ritonavir was present, and that level further increased in the amount by going from 3 months to 6 months. So these studies really are designed to push the formulation to the point of failure to really get an understanding of what can happen, and conversely, in other conditions where the samples have equilibrated to lower or moderate water content levels, this can help you to even define acceptable water content levels for shelf life storage of the product that keep it in a very stable region and with no observed conversion to crystalline ritonavir. There was effort on packaging assessment of the ritonavir powder. One option was to look at powder-filled capsules or bulk powder in bottles. And this was really shown that the powder is very prone to moisture uptake. It's quite hygroscopic, especially under the previously mentioned open dish or in-use storage conditions. Inside the capsule, the powder actually became encrusted, melted, or reverted back to a glassy film within a very short time period when stored under high moisture or higher temperature conditions. And that's illustrated in the picture at the bottom. In contrast, powder filled into foil laminate sachet packaging provided a very effective protection from water uptake, and the powder remained as a free-flowing powder within that package, even under high-stress conditions. This sachet commercial package configuration was selected then based on that very protective properties as a single unit dose product configuration. So the commercial package configuration in total includes now a foil laminate sachet, which was also assessed for child resistance and senior-friendly attributes in its final design. The sachet is intended to produce a suspension of 100 milligram in 10 milliliters of aqueous media. And this is prepared by adding the powder to the liquid vehicle in a mixing cup that is also provided. Doses less than 100 milligram can be administered as a specified volume, measured with a dosing syringe that is provided to achieve the appropriate ritonavir dose. For patients taking the even 100 milligram ritonavir dose, an alternative is that the contents of the sachet can be sprinkled onto a soft food where the entire dose would be then consumed. Our combination product strategy was a very simple one, to utilize commercially available mixing cup and syringe with minor modifications to customize the markings. We provide the cup and syringe with the powder-filled sachets in a kit. Our efforts on human factor studies were clearly not designed to further design the devices per se, but really to optimize our instructions for use to adequately guide patients or caregivers in preparation of the doses to be administered. Our commercial product definition resulted in a 100 milligram Ritonavir dose, supporting flexible dosing for primarily the PK enhancing doses that are known, with support of a dosing cup and a syringe. The dose can be administered as a suspension in water, infant formula, or chocolate milk vehicles, or sprinkled onto soft foods such as applesauce or pudding. The powder level in the sachet is just under 700 milligrams, representing a 100 milligram active dose. We provide 30 sachets per carton with a dosing cup and oral dosing syringes, and this is intended for global markets. In addition to the demonstrated stability of the powder itself, and primarily in the protective packaging of the foil laminate sachet, the Ritonavir oral powder was also shown to be bioequivalent to the Ritonavir liquid reference. And this was following administration with food. So now it's the same for the Ritonavir tablet and liquid, as well as the powder. With food, the exposure is actually reduced slightly compared to fasted conditions, primarily with respect to the Cmax. The vehicles that were qualified include, as mentioned, the water, infant formula, chocolate milk, applesauce, and chocolate pudding. And PK studies were conducted to show comparable bioavailability in all of those administration vehicles. With each PK study, we took the opportunity to also collect feedback on the palatability through questionnaires, where the subjects would complete the questionnaire within 15 minutes of dosing. The questionnaire evaluated characteristics of overall impression, taste, aftertaste, smell, and texture. And as shown below, on a scale of 0 to 9, there's a comparison here of the powder in water, in pudding, in chocolate milk, compared to the reference oral solution product. And in general, palatability scores across these different attributes are somewhat comparable. So there's no significant improvement with the Ritonavir powder, but there's also, it's not any worse than the oral solution. So in considering different options for now addressing palatability, it is known that there are different approaches to address drugs with inherently adverse sensory attributes such as bitterness. This is very well known for Ritonavir. It has a very inherent bitterness attribute. And our historical experience with the Ritonavir liquid formulation really showed that incorporation of sweeteners or flavors into the formulation were really not effective in addressing the lingering inherent bitterness of Ritonavir. So based on this, options to further modify the powder formulation to incorporate sweeteners and flavors was not viewed as a viable path forward. Rather, we looked at other opportunities that include dosing with food and having the concept of a food chaser that would be provided immediately following the dose administration. A variety of foods were selected based on multiple factors, including age, ease of preparation, and global availability. A number of these products are listed here, where in addition to the vehicles previously mentioned, we evaluated peanut butter, hazelnut, chocolate spread, fruit concentrate, syrup, honey, different options here that would be administered following the dose. A substantial reduction in the perceived bitterness was associated with foods that were high in fat content or sugar content, and this often correlated with low water activity and strong flavor intensity. In particular, peanut butter and the hazelnut chocolate spread were found to be most effective in quenching and reducing the inherent bitterness of the Ritonavir. Having studied a number of options really was aimed at providing choices for individual taste preferences for different patients. So in summary, through the development of the sachets containing Ritonavir powder providing a 100 milligram dose intended as a single-use presentation. This was really successful to meet current and anticipated future dosing needs for ritonavir in pediatric patients. The key design targets for the ritonavir powder were achieved. Specifically, there was complete removal of the alcohol and propylene glycol solvents, thus avoiding potential risks for infants and children. In replacing an already available oral solution product, we preserved the flexibility needed to meet dosing needs as a therapeutic agent, but more importantly as a PK enhancer. An acceptable dosage form as a suspension or sprinkle was achieved for pediatric patients. With the protective packaging and improved stability characteristics, we were able to demonstrate acceptable commercial shelf life under global climatic storage conditions, and this provides significant advantages through the supply chain, product distribution, and ultimately for patient use. Importantly, the ritonavir powder was shown to be bioequivalent to the ritonavir liquid and provides consistent performance in several liquid and soft food vehicles. And we've demonstrated opportunities for patients and caregivers to facilitate compliance and improve overall palatability through the use of various food products, commonly available food products, as a food chaser based on individual patient preference. With that, I will say thank you for your attention and ask if there are any questions.
All right, thank you very much, John. So, John, I see some questions popping up from my screen on taste mask formulation. So I'll start with two points here. So we have one question from Anna Jasinek and Lorraine Sequeira, so both on taste masking. So one question is, how did you assess the palatability at the start of your development of the enteric coated powder when you identified that there was no benefit of palatability with the coating?
So that was also done through questionnaires. Those were healthy adult PK studies, and through questionnaires we were provided the initial assessment to see if there was, on a comparable level with the ritonavir liquid, was there any benefit to the polymeric coating.
Okay, thank you, John. And to follow up on that, I think from Anne so looking into the formulation taste masking effect. So I think she's curious to know if the new formulation is taste mask. You have answered that, and I think working a lot with food products and chasers. And from the analytical perspective, do you see any challenges with analyzing the products when combined with food?
So there is definitely potential for some matrix interference when studying these with foods, but in all cases, I think we were able to overcome these to provide the necessary, to demonstrate the necessary in-use stability. The recommendation when mixed with food products is to administer within 2 hours, and that's primarily based on the food product itself being at room temperature for that time. There was no observed impact on the stability of ritonavir, chemical or physical, in that time period with any of the food products.
Okay, thank you. So we have one question from Henry Wu from Merck. I think he's interested to understand what studies were conducted to qualify the type of food and amount of chaser. First question. Second question is that how do you know if the chaser food has no impact of the PK of the granules?
So for all of the food products that were mentioned as administration vehicles, the various liquid vehicles and the soft foods mentioned, we actually did also perform PK evaluation in healthy adult humans to show that there was really no impact when administered with the food products. So we did do both the in-use stability assessment and the human PK assessment of those different vehicles. In terms of the food products for chasers, those were really done in through questionnaire or studied in taste panelists to assess the overall effect of the selected food chasers.
Thank you, John. And one more question from Yogesh Patel from Amgen to follow up on the compatibility studies. So is impurity testing a part of the compatibility testing with food?
Yes, we did assess for those compatibility, in-use compatibility assessments. We looked at both chemical and physical stability. So this included assay degradation products, as well as confirming that we maintained ritonavir in an amorphous state.
Okay. Thank you, John. One more question from Janice Lee. You have shared that a significant number of pediatric patients are in sub-Saharan Africa, and the reason for reformulation of ritonavir. So what is the plan to introduce this formulation in sub-Saharan Africa? Currently, it is still not available although in Europe, it has already replaced ritonavir solution.
Correct. So those global submissions are ongoing, and in some cases have occurred and are under review. So our intent is really on a global scale to provide the ritonavir powder in markets where currently it's either ritonavir liquid or perhaps no alternatives at this time. But I would say that in particular to those areas in South Africa, the intent is to pursue regulatory approval and provide the product in that region as well.
Thank you, John. And we have a question from Mike B. from Amgen. So Mike's interested to know how many years does it take to develop the final commercial formulation, and what level of resources were required?
So development of the ritonavir powder, I would say certainly leveraged all of the extensive experience that preceded it. And I think that's important to acknowledge that had we started from scratch, I think the effort would have been quite long and quite significant to ultimately arrive at the amorphous solid dispersion formulation. Much of that foundational work had already been completed with the respective adult products. So with that as a significant starting point, to take that further into a suitable pediatric presentation was still, I'd say, took several years, and I'd say is still ongoing in some respects, really with continuing to optimize from a human factors perspective the instructions for dose preparation and administration.
Thank you. So, John, so Janice Lee has a follow-up question to your answer on the sub-Saharan Africa strategy. So she would like to know if you have further the same plan for the lopinavir ritonavir solution.
There are no current activities, no plans at this time. I think in monitoring, I do believe that there are other lopinavir ritonavir options for pediatric that we are aware of as being either available or soon to be available. So we're monitoring that progress as well, but there are no current efforts to develop a similar formulation for Kaletra.
Right, thank you, John. And any other questions from the floor? So if not, I will say thank you, John, Dr. John Morris, for sharing his experience on ritonavir. And just for the benefit of the webinar series and ways to improve series in the future, we would like to invite you to participate in a survey. Mahalia will send the survey out by today. So please, for those participating in the rooms organized by the IQ members, please, we appreciate it very much if you can approach your IQ members for the survey link and join us in the survey to help us improve the webinar better. And one last point to share is that in next month, we will have extemporaneous drug preparation presentation by Karen Thompson from Merck. And please join us in the session next month at the same time on the first Thursday of October. And again, thank you very much to John, and thank you everyone for participating.
Thanks very much, David. Thank you all, and I hope you have a great day.
All right, thank you, Mahalia.
Thanks. Bye-bye, all.
Bye-bye.
Thanks.
Bye-bye.